In this topic, we're going to have a look at dihybrid inheritance. So by the end of this topic, you should be able to answer the questions: how do we do a dihybrid cross? What is the law of independent assortment? What is the expected ratio from a dihybrid cross between a heterozygous parent and a homozygous parent? And what is the expected ratio from a dihybrid cross of two heterozygous parents? So Mendel's experiments involved pea plants. We're going to look at how two characters determined by two genes located on different chromosomes are inherited. This is dihybrid inheritance. Gregor Mendel investigated the inheritance of two characters of pea plants at the same time. These were the seed shape and the seed color. So he looked at the round shape, which is dominant, compared to the wrinkled shape. And then he also looked at the yellow colored seeds, which are dominant, to green colored seeds. So his experiments involved crossing two pure breeding plants, one that produced round-shaped yellow-colored seeds, and the other that produced wrinkled-shaped green-colored seeds. So before we can do the dihybrid cross, we need to write the letters for each allele. Since the allele for the round shape is dominant, we represent it with a capital R. The wrinkled will be a small r. We're going to choose the letter G for color because we're not choosing Y because the capital Y and the small y are very similar. So capital G will represent the allele for yellow and small g will represent the allele for green. So when you do a dihybrid cross, the first step is to write the phenotype and genotype of the parents. So the phenotype will be round yellow and wrinkled green. Because they are homozygous, the genotype will be capital R, capital G, and small r, small g, small g. The next step is to write the alleles in the gametes. So you have capital R, capital G in one gamete, capital R, capital G in another gamete, small r, small g in two other gametes. Then you use a Punnett square to work out the F1 generation. So what you do is you write the gametes at the top, capital R, G, and then small rg on the left, and you cross these together. You'll notice that all the seeds will be round and yellow because capital R represents round and capital G represents yellow, and those two are dominant. So your F1 generation genotype will be capital R, capital G, small g. So the phenotype will be round yellow, and your ratio will be all of them are round and yellow. Now if you allowed the F1 generation to self-pollinate, that would mean that you've got two heterozygous parents crossing. So what's the first step that you do? You write down the F1 generation which has been self-pollinated, phenotype, genotype. So it's round yellow, capital R, capital G, small g for both of them. What's the next step you do? You're going to write the gametes. So remember to put them in circles. Capital R, capital G, capital R, small r, capital G, small r, small g. This is so that all the alleles are represented in the gametes. Then you use a Punnett square. So you write down your gametes at the top and the gametes going down the sides, and then you cross them. Once you've done that, you're going to notice or look at the different colors of the seeds and the shapes. And you're going to notice a ratio. So count up how many round yellow seeds there are. You should count 9 round seeds. What about the number of green round seeds? How many are there? 3. And yellow wrinkled seeds? 3. And then how many wrinkled green seeds are there? Only 1. So you're going to write down the F2 generation phenotype. So all the different phenotypes that you've got, round yellow, round green, wrinkled yellow, wrinkled green. And then you write down your ratio. 9 is to 3 is to 3 is to 1. Okay, 9 to 3 to 3 to 1. This ratio is very important to remember. Which one has only got a ratio of 1? It's your recessive, homozygous recessive. So if we were to cross a heterozygous parent with a homozygous recessive parent, what do you think you're going to get? Well, what's the first step? The first step is to write down the parents, phenotype and genotype. So the phenotype will be round yellow, and the genotype will be wrinkled green. Then you're going to write down the gametes. You're going to use a Punnett square to work out the recombination. So you will notice that you should get a ratio of how many round yellow, 1, how many round green, 1, how many wrinkled yellow, 1, and how many wrinkled green, 1. So your offspring will have a phenotype of round yellow, round green, wrinkled yellow, wrinkled green. And the ratio should be 1 is to 1 is to 1 is to 1. So you should keep these ratios in mind. The other one was 9, 3, 3, 1, and this is 1, 1, 1, 1. Okay, looking at the law of independent assortment. As chromosomes arrange themselves randomly on the equator during meiosis, any one of the two alleles for the seed color can combine with either of the alleles for seed shape. The result is that you get 4 different gametes. So the law of independent assortment states that each member of a pair of alleles may combine randomly with either of another pair of alleles. So what do I mean by this? Well, here's a diagram to explain what I've just said. You can see that the chromosomes are arranging themselves randomly on the equator during metaphase 1 of meiosis. Any one of the two alleles for seed color can combine with any one of the two alleles for seed shape. The result is that you're going to get 4 different types of gametes. So here you can see how the round and yellow during anaphase go to one pole and the wrinkled green go to another pole. So the result will be that you have one gamete with yellow and round, another gamete with green and wrinkled. If you had another arrangement where you had wrinkled and yellow on one side and round and green on the other side, what's going to happen? During anaphase, they're going to separate towards different poles, so that you're going to get a gamete with yellow wrinkled and a gamete with green round in it. So this explains independent assortment. Now, variation can also be increased if these different gametes fuse with gametes from another plant. Okay, in tomato plants, there's a gene for stem color and shape, and both of them have got 2 different alleles. So you've got an allele for a purple stem and a green stem, and an allele for a jagged leaf and smooth leaf. Looking at the letters that I've used, which allele is dominant for the shape of the leaf? It's the jagged leaf. Which allele is dominant for the color? It's purple. Okay, so both genes are on different chromosomes and are sorted independently at meiosis, what we've just discussed. So the genotypes are going to result in different phenotypes. Having a look at these different genotypes, this is the phenotype that you'll get. Okay, so notice how the purple stem is dominant, so we represent that with a capital A. The cut leaves, capital D, is also dominant. So wherever you see a capital A and a capital D, the color will be purple and it'll have cut leaves. If you have a small a and a small a together, it's going to result in a green stem, and small d will be smooth leaves. So let's do a question. What is the expected ratio of offspring if you cross two parents that are heterozygous for both characteristics. So looking at heterozygous parents, the first step is to write the parents' phenotype and genotype. So purple cat will be capital A, capital D, small d. Purple cat, capital A, capital D, small d. Then you're going to write the gametes. What's the next step that you're going to do? You're going to use a Punnett square. So you're going to cross the gametes, the four gametes with the four gametes. You're going to write them across the top and across the side. You're going to cross them together and you should get a ratio. What ratio can you see? Count how many purple jagged leaves there are. You should find 9. How many purple smooth leaves are there? 3. How many green jagged leaves can you see? 3. And how many green smooth leaves are there? 1. So your F1 offspring's phenotype will be purple cut leaves or jagged leaves will be 9. Purple smooth will be 3, green cut 3, and then green smooth will be 1. So what is the expected ratio of offspring of a dihybrid cross between heterozygous organisms? It should be 9 is to 3 is to 3 is to 1. Now remember that there are 16 different possibilities. So if you were to add those up, 9 plus 3 plus 3 plus 1, it should add up to 16. And we're going to look at something called a chi-squared test later on, which can be used to show any differences between experimental observed results and the expected ratio. So in summary, what is the law of independent assortment? Each member of a pair of alleles may combine randomly with either of another pair. What is the expected ratio for a dihybrid cross between a heterozygous parent and a homozygous parent? It's 1 is to 1 is to 1 is to 1. What is the expected ratio from a dihybrid cross of two heterozygous parents? It should be 9 is to 3 is to 3 is to 1. And that concludes our lesson. The end.